Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. Today we will talk about how to calculate atom distances in PyMol. What specifically we will talk about in this session, have a look. We will cover two basic stuffs or two basic points. First, we will learn how to calculate atom distance in structures contains two or more proteins. So, in what scenario you may have these structures? So, for example, you perform some protein-protein docking. In that case, you have more than one protein structures or more than one protein chains, I would say, in your PDB structures. In second scenario, sometime you download a structure from the PDB and it contains more than one proteins. Third scenario, you download a structure from PDB and it contains oligomeric status of protein. From oligomeric status, I mean it may be dimer, trimer, tetramer, pentamer, and so on. So in point 1, we will learn how to calculate the distances between different atoms in this assumption or in this scenario I discussed already with you. And we will also discuss the limitations in these in the dimer or trimer specifically. So, okay. In point 2, we will discuss how to calculate atom distances in protein and ligand. You will be able to find information on YouTube from different people about the point 2, but very few people have covered this point. So our main task is this, but then I realized that why not to cover both of them. So today we will cover both of them in a short time. And the last thing we will use PyMol. Why I use PyMol? Because I have covered its installation, its publication quality images, tutorials, and all these. And also, I believe it is one of the most state-of-the-art software for publication or for generating publication quality images. In today's tutorial, I will use a PDB structure 4EXS. You can download for yourself if you want to follow this tutorial exactly. And why I choose this structure? So it has the reason. This structure contains two symmetric chains, A and B. Okay. Also, it has ligand in each chain. So, dealing this protein, we will also learn how to handle the chain problem if your protein has this kind of problems, and how to handle residues which are represented twice in a structure. For example, in 4EXS, there are two chains and I use the word symmetric. So in symmetric structures, it's a kind of dimer. So there you will see the residue twice in two different chains because it's actually the pictures of the two proteins in different crystal unit. So let's continue. I will close this and we will start the PyMol. Okay, so here we are. I'm going to start PyMol. I have installed. This is my PyMol. I will go and I will keep the display white, so that's okay. Now the structure I have already downloaded and it is present here uh, in the download folder. So I will load this structure first. It is present in download, so 4EXS is the structure. Now, let me make it bigger. Okay. Now, if you see, I come here and color by chains. So you can see two chains in different colors. So now, if you are following this video because you have a protein structure or a model and it has two chains, so exactly this protein also has two chains. But this protein offers an extra challenge. The challenge that offers, excuse me, okay, the challenge that offers, if you see, like this one is residue 42, GDQRF. If you see here in this chain, GDQRF. So I means both the structure that contains the same residue twice. If you want to probe a residue 42 in one chain, so it will not be as simple because we have to use the chain identifier residue 42 of chain A or chain B. Okay, so let's start and I hope you will follow me. Okay, I will try to explain in a basic way. I will use these 7 commands and I will also teach you how you can use this command in your case. From where you will find these commands, I will drop them in the description section of this video. Please copy and make your own file in Word or text file for yourself and store it in your local computer. Okay, so first we are going to exploit this command. Okay, protein structure. So if you are dealing protein structure, so what we will do, if you see or if you look to this command, it says select string name, comma, forward slash, your protein, double forward slash, A, 42 and CA. Please understand first. The second command is also similar except A is changed to B. Please understand first. You cannot change the select term. It will be as it is. For string name, you have to change it. So we will change it. You will see. Then comma forward slash your protein name, which is loaded in the PyMol. Then which chain you are talking, A, B, C, D, whatever, which residue you are interested, this one, and which atom you are interested. Now, in case you don't know to what are those atoms in my proteins or in my uh, in the atoms in the amino acids, I mean that. Okay, what we will do, we will use the iterate command. For the iterate command, you don't need to do anything, just copy and paste it here. What this command will do, I will tell you. Just first do it. Okay. It has generated a long string. You can see long strings of information. But the beauty of these information is that now you are able to see each and every atom of each and every residue, including the water and including your ligand atoms if present. For example, let's talk about THR, nitrogen. Its number is 34 and it is included in chain A. Again, THR, then the carbon alpha, Ca is for carbon alpha, 34A. Let me highlight it. So this is for residue 34. If you see nitrogen connected to carbon alpha, carboxylic carbon, then the carbonyl oxygen, then the carbon beta and the carbon gamma 2 and the oxygen gamma 2. Okay, the carbon beta is the one which directly connects to the carbon alpha, and in simple terms, we call it R group. Okay. It will also help you if you have a ligand. So just come down. You will see the ligand information in the bottom. You see when here you can see X at Z. So how many atoms this X at Z contains? It contains C1, O1, C2, O2. The same way it will print. This is for the beta chain, and the same or for B chain, I would say, or and we because it is a symmetric structure, so it should contains two two units. So this is the ligand atom of chain A. These are the water of chain A. These are the zinc atoms which are present in the structure of chain A. So this iterate command will print each and every atom for you, so you will not make a mistake, or it will minimize the chances for a mistake. Okay. Now, okay. The second step is, now we are going to do some steps for protein structure. Let's suppose you have protein structure which has more than one chain, and you are interested in to calculate the distances between certain residues. But for sure, every residue contains some atoms. Normally, we are calculating the distance between two residue, and we use carbon alpha as a reference atom. We also call it heavy atom. So normally we use carbon alpha. So today I will also use carbon alpha. But if you want to use some another atom, so you can use it because all the informations are printed by this command. So you can use another atom. Just change the Ca here. Okay. I will copy this first and will come to this. So now you can use this part, or okay, I will use this part. So just control V, don't click enter. We have to change something. Okay, let's say I am interested in the residue 43. So I change this 43 of chain A, and which atom of the residue port 43? Ca, carbon alpha. What is my protein name? Just change it here. It is 4EXS. You can check it here. Okay, now the string name, I will say like 43 underscore A. This is my string name. When we run it, you will see it here 43 underscore A. Just say enter. Okay. Can you see it says one atom selected? It's here if you can see a red dot. Okay, and you can see here. That's okay. Now we copy or paste the command again. Now we are interested in, let's say, the same residue 43, but in chain B, not chain A. Okay, and the protein name is the same 4EXS, and the string name is 43 underscore B. Okay, now enter it. So we have two strings generated, it's here. So we will see a distance between this point and somewhere here. Okay, now we have two selections enabled, 43A, 43B, and before creating a selection, you just take care of this term, that is there anything selected or not. If it says zero atom, it means you are making some mistake. So be sure the program should say one atom is selected. Okay, now we will use this thing. copy and paste it here. Okay, don't change the distance. Now we will change the string name. So a string name, our string name will be say 43, sorry, 43 INTR. It means 43 interaction. Okay, between we want to check the interaction between two things. The first is 43 underscore A. So just change this way, 43 underscore A, and then don't remove the comma, 43 underscore B. Okay, we have created these two things already. Now we are going to use the distance command, and the new entry will be called 43 underscore intr. It will appear here. So let's enter. Okay, can you see this? 43 intr. Now it's almost 70.6 inch strong. Okay, so now if you want to calculate more interactions, and it will be added here during the time when you generate a figure for your manuscript or thesis. You can turn off the interaction, or if you think no, I need to turn it on, if you have many, so just turn it on. So it will give you a greater flexibility. One thing more, if you worked so hard for generating something, and let's say abruptly someone asks you that go and let's go outside, then what you have to do, just save session. So it will save the pse session.pse. So next time when you open PyMol, just load the pse session and you will start from there. Okay, this is another trick I showed you. So it will not waste your time. So this was the way how to calculate the distance between protein residues. Now we are quickly going to calculate the distance between ligand atoms and a protein residues. So we have two chains. This one is one chain, this one the cyan color and the green color is another chain. Every chain contain its own ligand, like this one contain here, if you can see here, and this one contain here 4xe8. So it's up to us what we are going to do. So okay, I will select one chain A, and I will choose some residue and some ligand atom, and will generate a distance, or you can also call it interaction between them. So once again, if you understand this video so far, so we will move here, protein ligand structure. Okay, so just copy, and let's say I have copied it. Okay, string name, I will say, let's say ligand, no, let's say res 43. I'm using the same residue. Let's say 44. Okay, protein name is 4EXS, 4EXS, chain A. Okay, and here also change it to 44. This one, res 44. Now we will choose the ligand atom. The ligand atom we have already printed, so you can check it there. I will choose directly the S. Again, the select, let's say ligand, and then S, let's say underscore S. And your protein name is 4EXS. Ligand, okay, you have to change this LIG. So where you will change it? Just see the sequence, the easy way. you can see the ligand name. If it is LIG, leave as it is. If in my case it is X8Z, or if your case is different, so just change it to that name. So I will change it to X8Z, and the ligand atom is S. So actually I want to calculate the distance between S and the residue 44. So okay, it it says zero atom selected. It means I have made some mistake because I need to identify a chain, because there are two X8Z. So I will say here A and do it now. Okay, now you can see one atom selected. Okay, I will drop a correct code in the description section, so take it, check it from there. Now we have two strings generated, res44 and leg underscore s. Now we want to calculate the distance, so just copy this. First, our new string will be, let's say, Protein-Ligand, and the first string we generated is res44. The second string we generated is lig underscore s. Okay, can you see? It's here. It's generated from there to there. Now, if you want to turn both the interactions on, or if you have many, let's say, so you can see I have on both the turn on both the interactions. Okay, I hope you will like this video, although it seems a basic video, but I'm sure people are finding it, few people are struggling doing this kind of job. So I hope from now on you will be able to do this kind of things. And if you want to generate a beautiful or impressive figures, I will also share the link in the description of my previous videos. You can go there if you are unable to install PyMol. So I have already shared for Python 3.10. So you can install it, and if you need some wheel files, please drop in the comments. I will provide you. Thank you so much, and if you have any question, you are welcome to ask. Bye bye.